Okay, ladies and gentlemen, we're going to start today. It's been quite a while since we had our annual meeting of the Genetic Society of Israel. So welcome all members and guests of the Genetic Society of Israel to our annual, and this time not meeting, but a symposium. For those of you who do not know me, I am Dan Mishmar from Ben Gurion. I'll be your new chairman of the Genetic Society of Israel after a long term that Martin Kupiec served as one and as such. So today, in the spirit of our time, the symposium is being held online in Zoom. We are about to hear two young Israeli scientists, wonderful scientists, in my opinion. They are both new PIs, Michal Rabani from the Hebrew University and Benjamin Rosenthal from BGU. After the business meeting, we're gonna hear an established PI, Jonathan Weissman from MIT. Now, as this is an annual meeting of the GSI, we're obligated to have a short business meeting in which we will present the new board members and matters that require approval by the entire membership. So do not worry. It will be short and it will be in between the talks, namely after the talks of the two young PIs and just before Dr. Weissman's talk. So please stay for this meeting. This is important. We need you there. So without further ado, let's start with Michal Rabani. So Michal Rabani came all the way from the Hebrew University directly to this Zoom meeting. Please, Michal.
Thank you. Hi, everyone. Very happy to tell you today about our work on understanding the sequence element that regulate maternal mRNA degradation in the zebrafish embryo. So I'm going to start with development, which is really one of the most amazing transformations in biology, where a single cell fertilized egg transforms into a whole organism with many different cell types, tissues, and organs. And one of the basic processes that enables this transformation is that embryonic cells divide, and then each of them differentiate into a different cell type. And that process happens by developmental transformation, in which these cells change their RNA content. And to change their RNA content, there are two parallel processes that happen in the cells. First, they degrade the old RNAs, and at the same time, they also make new RNAs that replace them. Now, we already know a lot about how cells make new RNAs, but what we understand much less about is the role of RNA degradation during developmental transitions. But what we do know is that RNA degradation is crucial for developmental transitions. And one of the most striking examples for that is one of the earliest developmental transitions that happens in all animals, and is the maternal to zygotic transition. So here you can see the development of a zebrafish embryo. In the early stages of development, the zygotic genome is silenced, and all functionality is achieved by maternally deposited RNAs and proteins. But after approximately 3 hours, when the embryo is almost 1,000 cells, there's transcription of new zygotic RNAs that gradually replace the maternal mRNAs, and there's a massive degradation of the maternal mRNAs. And we know that this degradation of mRNAs is really crucial for normal development. So, for example, there's one very well-established pathway for the degradation of maternal mRNAs in zebrafish that is happening by the regulated by microRNA, miR430. So this microRNA is expressed from the zygotic genome after it's activated, and it can recognize and bind specific sequences on maternal mRNAs and lead to their degradation. And it was shown that when we interfere with the activity of this microRNA, there are some developmental consequences to that. But regulation of maternal mRNA degradation is not a simple process as this example of MIR430 would imply. In fact, when we look at the degradation of different maternal mRNAs, we see a lot of different degradation patterns. Some mRNAs are degraded slow, some are degraded fast, some are degraded early, and some are degraded later after the activation of the zygotic genome. So our research question was to try to understand how this information that encodes all these different degradation patterns is encoded in the mRNAs, in the maternal mRNAs. And this question has important implications both for fertility, because degradation of maternal mRNAs actually starts already during oocyte maturation, and of course for development, as would be implied by maternal effect genes. We wanted to understand how degradation of maternal mRNAs is encoded, and our hypothesis was that just like MIR430, there are different regulatory elements and different factors that can bind them. And the combinatorial effect of the different factors that bind each mRNA generate its unique degradation pattern that we observe in the embryo. But in order to map all these diverse regulatory pathways, we wanted to develop a new method. And for that, we developed a method that we call UTR-seq. So UTR-seq is based on a very simple principle. If we want to understand a regulation by a sequence, for example, the 3' UTR of this particular gene, we can take a fragment of the 3' UTR, clone it into a reporter, make mRNA from that reporter, and then inject those mRNAs into an embryo. The embryo will develop, and we can measure how much we see of that particular reporter during development. For example, here you can see that the amount that we measure at the early stages is almost the same as what we measure in later stages, suggesting that this reporter is in fact stable. And we can also show that on a graph where we plot the RNA amounts that we measure over time during development. Now we can do that with another sequence, take a different part of the UTR, clone that again into a reporter, and if we're lucky now, we see a completely different degradation pattern where after 10 hours, we have only very few molecules of the reporter left, suggesting that now the degradation is much faster. And that gives us a hint as to what kind of sequences to look for and where on the UTR to look for. But can we now take this approach and apply it more systematically? So to do that, we use massive parallel reporter assay. We take all maternal mRNAs, or at least those that are expressed at significant amounts during early development. There are a few thousand of them. And we look at their annotated 3' UTR sequences. And now we break those 3' UTR sequences into short and overlapping 110 bases long fragments. And we do that for each and every one of those thousands of maternal mRNAs and generate a set of 90,000 endogenous 3' UTR sequences. Each of them is 110 bases long. And it is very possible today to in vitro synthesize all those different sequences and get them in a tube, basically. And now we can take all the 90,000 different sequences and clone all of them into reporters and make a library of mRNAs that has one of 90,000 different sequences in their 3' UTRs. And all those 90,000 sequences are pooled together. And we inject them all together into an embryo and basically do the same thing that we do with a single UTR, only with 90,000 of them, and use RNA sequencing to measure the amount of RNA that we have from each and every one of these 3' UTR sequences. And indeed, when we do that, we again see a variety of degradation patterns in those reporters. So some are degraded fast, some are degraded slow, early and late. And overall, we can divide these reporters into two main degradation patterns. The majority of reporters, almost 80% of them, are degraded immediately after fertilization, and degradation progresses at a constant rate. But for a smaller fraction of these reporters, they are initially very stable, and their degradation only starts after the activation of the zygotic genome, approximately 4 hours after fertilization. And we also see some differences in the average degradation rate in each of those groups. And that made us hypothesize that we're looking here at two different degradation pathways. A maternally encoded pathway that is active already after fertilization, and a pathway that depends also on some zygotic factors that are only expressed after the zygotic genome is activated. So to verify that, we took our reporter library, which we studied in embryos, and we also injected it into unfertilized oocytes. These oocytes contain all the maternal factors, but they don't contain any of the zygotic factors. And indeed, in the oocyte, we see that early degradation happens almost at identical rates to embryos, but late degradation doesn't happen in oocytes. Now let's go back to our original question and try to use these reporters to understand how the sequence of the mRNAs affect their degradation. And what the reporters allow us now to do is to simplify this question and basically ask how the 3' UTR sequence of a reporter, which is only 110 bases long, is connected to the degradation rate of that particular reporter as we measured it in embryos. And what we did to understand that is that we developed a computational model, a linear regression model, that can convert sequences into degradation rates. And what this model does is it finds the weight of each short sequence and how much that particular sequence would affect the overall degradation of a reporter that contained that sequence. So let me try to explain that with a bit more details. So we have a regression model, and for each short sequence that is 3 bases to 7 bases long, the regression model gives a weight, a number, how much this particular sequence contributes to degradation. So now if we have a new 3' UTR sequence, we can break it down into all the short 3' nucleotides, 4 nucleotides, up to 7 nucleotide long sequences within it. And we can sum all the weights of those particular sequences to get a predicted degradation. And this is basically a code that defines the rules for sequence-based degradation in our reporter library. So what did we find? So basically this approach identified 6 key regulatory elements in the 3' UTRs of the reporters. 3 of them are associated with the early degradation pathway, and they can be either stabilizing or destabilizing. And then there are 3 additional sequences that are associated with late onset degradation. And these are destabilizing elements, including the MIR430 SID sequence, which is already a known mechanism. And so now can we see that this model actually works? How can we do that? So what we did is that first we compared the degradation rates that the model predicted with the degradation rate that we actually measured, and we showed that the predicted rates recapitulate most of the variability that we see in the experiment itself, which is a nice first validation. But then we did something else. We took specific 3' UTR sequences that contained elements which the model predicted to have a strong effect on degradation. And we used the model to modify them in a way so that they no longer contribute to degradation. Or we took sequences that did not have any elements with a significant contribution and used the model to add new sequences that are predicted to have a significant contribution. Now we took these new sequences, we injected them into an embryo, and we validated that indeed we see the change in degradation as we expect to see by the prediction of the model. So I showed you how we developed a massive parallel reporter assay that we call UTR-seq that can measure the degradation of tens of thousands of mRNAs in parallel within an embryo. And we developed a computational regression-based approach to predict degradation rates and sequence elements from this library. We identified two main temporal degradation programs: an early onset maternal degradation and a late onset degradation program that accelerates degradation of maternal mRNAs after the zygotic genome is activated. We identified 6 key regulatory elements, and I didn't show you that, but we verified that they also have an effect on endogenous maternal mRNAs. And we used our model to design artificial 3' UTRs with specific degradation. So now in the last few minutes, I want to briefly show you some new results that we got that address the question of the physiological relevance of maternal mRNA degradation and how is that important for development.
Michal, 2.5 minutes.
Okay, I'll be done. So Daniel Segal is a master's student in my new lab. And he took a maternal mRNA called buckyball, or BUC in short, that is expressed in zebrafish embryos. Now, buckyball is a prion-like, encodes for a prion-like protein that creates RNA aggregates in the embryo. And these aggregates that are formed very early already in the 2-cell stage in the embryo, in the cytoplasm of the blastomeres, is responsible for the differentiation of primordial germ cells for the zebrafish embryo. So as you can see, the mRNA of buckyball is very unstable, and immediately after the activation of the zygotic genome, we see a fast degradation of this mRNA. But Daniel also identified another maternal mRNA of a different gene, MAGO, which is very stable in the embryo. So what Daniel did is to take a BUC GFP reporter that we had in the lab and replace its 3' UTR with the MAGO 3' UTR. Now this completely changed the regulatory elements of the mRNA, because in the original MAGO 3' UTR, we had a MIRF30 element that lead to destabilization and degradation of this mRNA. Whereas in the new MAGO 3' UTR, we had more than 6 copies of the stabilizing poly-U element that makes this mRNA much more stable. And indeed, it's maybe not surprising, but when Daniel overexpressed this new construct of BUC with the MAGO 3' UTR, he saw a significant increase in the stability of the mRNA in the embryo, and also a significant increase in the expression of the BUC protein in those embryos. And that had some physiological effects on development, whereas when you inject the overexpress the BUC with the original 3' UTR, embryos develop more or less normally. We see much more severe developmental problems with the destabilized version, including the ectopic formation of primordial germ cells and other more general physiological effects, which we are now following up on. So to summarize, I showed you a massive parallel reporter assay that can help us to understand the sequence-based rules of maternal RNA degradation. And it's also a general tool that can help us design and engineer mRNAs and predict the effect of genetic variation to understand the physiological implications of RNA degradation on development, and is also broadly applicable for other biological functions and systems. So the first part of this work was done during my postdoc in Alex Shear lab, and now I have my own lab at the Hebrew University. And if any of this is interesting to you, you're welcome to contact me. Thank you.
Beautiful, Michal. Thank you so much. We have time for two questions or 3 quick ones. So please use your hand.
I have a question.
Who's asking?
Yes.
Can I ask a question?
Yes, you
So
can.
the question is there any effect to the length of the poly-A stretch?
Yes, the answer is yes. we already know that MIR430 changes the poly-A length. And we are actually developing now a new reporter assay that measures directly the poly-A length of the reporters after we inject them into the embryo. And we see that almost all of these elements affect the poly-A length of the reporters. So probably a lot of the effect is mediated via the poly-A.
Thank you.
Any more questions? We have time for another question.
I have Michal.
Yes.
Beautiful work. How conserved are these motifs? Because any change will change the developed organism.
That is true. Conservation is actually not that high, but it could also be because the positions are changing and the overall UTR sequence is not very conserved. So comparison of sequences is a bit more challenging in this context. But these are very well-known regulatory elements of RNA stability. So MIR430 in itself is not conserved, but the other elements that we found are actually very conserved up to humans and mammalians in humans.
Michal, I'd love to ask more questions, but we need to move on. Thank you so much, Michal.
Thank you.
You're welcome. So let's move to our next speaker, Benjamin Rosenthal from BGU. Could you please share with us your talk? Thanks, Benjamin. You can unmute yourself so we could hear you.
Can you hear me now?
Yes, we can.
Okay. First of all, thank you very much for having me here. I'm from Ben-Gurion University at the Department of Microbiology, Immunology, and Genetics. I'm an immunologist, and today I will tell you about a unique marine model organism that we use to study innate immunity-based histocompatibility and transplantation. I would like to mention that a lot of the work that I will be presenting was done during my postdoctoral research at Stanford University with Professor Irving Weissman. Well, what does it make a Botrytis lesseri a unique model to study innate immunity-based transplantation? And I will tell you, the Botrytis, which is part of the tunicates, on the evolution, it's our closest invertebrate relative, which is one step in evolution before the evolvement of the adaptive immunity, which means that all of its responses are innate immunity-based. But I think the most interesting feature of the Botrytis is the natural transplantation phenomena. And I would explain. When two different individuals are coming together and touching with the ampulles, the end point of their blood vessels, there can be two outcomes. Either a rejection by creating these necrotic areas between the ampulles, or a fusion. And the fusion is happening when the blood vessels are coming together and fuse into one blood vessel. So we have cells freely going from one animal into the other. So we have also this natural transplantation through the fusion. And we also have stem cells now going from one animal into the other. And those stem cells are now competing who will build the new soma or the gonads of this new chimeric animal. So we have also natural transplantation, and it's also stem cell based. So what I was interested is in understanding what are the cellular immune mechanisms enabling this fusion or rejection and this recognition. So the first thing we have done was developing immunological functional assays. So the two main cellular functional functions of the immune cells, it's either phagocytosis, engulfment of a target cell, or cytotoxicity, a direct killing of a target cell. What we have discovered that while there's no difference between self-compatible or compatible and rejecting colonies in phagocytosis, we saw a significant upregulation in cytotoxicity where we have rejecting colonies, suggesting that cytotoxicity is the mechanism of rejection, just as we have in mammals, where we have natural killer cells and cytotoxic T cells, where we have a rejection of transplantation. we wanted to validate this. So we differentially labeled cells coming from the different colonies to see the allogeneic phagocytosis. We see the green cells engulfing the red ones and vice versa. To validate cytotoxicity, we have done different effector to target ratio, the same way we do with T cells and NK cells. And as you can see here, we have a specific lysis in rejecting, again, showing that cytotoxicity is the mechanism of cellular rejection. But the question is, can it happen in vivo? This is something that we see in the vial. So for that, we have developed in vivo transplantation assays by differentially labeling the animals before they come together, either in a fusion of the blood vessel or in rejection and creating these points of rejection. And what is very interesting, that when we look in the points of rejection, we discovered that the cells originated from the different colonies are mixing up and killing each other, suggesting that this cytotoxicity is taking place in vivo. I would like to emphasize that this assay is done on a live animal utilizing the transparent body of the Bacillus. Utilizing the transparent body of the Bacillus. So we can actually now follow these allogeneic cells in the transplantation, the green cells going into the red animal and vice versa. We can follow where they're sitting down, and we can also take the cells to flow cytometry and analyze the level of success of the transplantation. Well, I showed you ex vivo assays, I showed you in vivo assays, but the question is, what are the different cellular populations that can recognize their targets? So for that, we use the flow cytometry sorting. We isolate cells from the Bacillus, and then we analyze by flow cytometry. This is without any labeling, just using size and granularity and autofluorescence of the cells. We can isolate about 10 different cellular populations, but some of them are still very heterogeneous, so we wanted to go deeper than that. So the big question is, what do you do when you have a non-classical model organism and you want to create clusters of differentiation, like the CDs we have in humans and mice? So I'm gonna make a very long story very short. We screened more than 130 different markers that could differentiate cellular populations, like antibodies that we screened by cyto, lectins that will bind to different sugar groups, and also enzymatic activity that we can recognize different parameters of the cells and analyze different populations. And eventually we have created a panel of 34 different cellular populations to sort them out. And just to give you the idea, this is Bacillus cells unsorted before going to the FOX, and this is some of the populations of our panel. So this is the panel we have created by flow cytometry of 34 different populations, and this is the parameters that we use to separate. Still, we used also size and granularity and autofluorescence. But isolating the cell, this is number one. What about the gene expression of the different cellular populations? So for that, we have done RNA sequencing of all the different populations, and we analyze the genes. And here you can see a heat map of the top 500 genes to separate the different cellular populations. And what is very interesting, that the populations that are different on the morphological level by flow cytometry are also different on the gene expression analyzing the RNA-seq. And now we can look on different populations and clusters of them. And I will give you an example of this cluster of population 33, 34, and 25 was very interesting to us. And why it was very interesting, because the genes that were significantly upregulated and annotated to mice and human genes are associated with the hematopoietic system. And this is a tool that was created in the lab, a gene expression commons tool. This is a mouse hematopoiesis model. As you can see, it starts with the hematopoietic stem cells and precursor cells, the lymphoid lineage where we have T cells, natural killer cells, B cells, the myeloid lineage, including granulocytes and monocytes, red blood cells, and platelets. And when we took the 235 genes significantly upregulated and annotated and put on it, we see a significant activity of the hematopoietic stem cells and immediate precursors, and also the myeloid lineage. I would like you to remember this, that we found the hematopoietic candidate on the molecular level, but right now I would like to talk about the immune cellular populations. Due to lack of time, I will just summarize it, that we did succeed in finding the myeloid lineage and also the phagocytic populations, and also the cytotoxic cells that are rejecting the tissues. And what we found is that the Bacillus as a tunicate is somewhere between the vertebrates and the invertebrates. On one hand, it has a true myeloid lineage, homolog to the vertebrate myeloid lineage, but it also has large phagocytic cells and amoebocytic cells, as we find in other invertebrates. We found that cytotoxic morula cells, and we assume that other invertebrates also have similar cells. We have some candidate for lymphoid lineage, but we did not test it on functional level, which is still leaving a huge question mark. It's what is the source of the lymphoid lineage or the source of our own adaptive immunity? Unfortunately, at this point, I will leave you with that question mark, and I will take you back to the discovery of the hematopoietic stem cells on the molecular level. So we wanted to test them. So the first thing we have done is homing testing to the known stem cell niches within the Bacillus. So the two known stem cell niches, one is cell islands, which is more gonadial stem cell associated niche, or the endostyle, which is more a somatic stem cell associated niche. And what we have discovered that our candidate hematopoietic stem cells are localized to the endostyle somatic stem cell niche. So we have molecular level, we have localization to the stem cell niche, but what about differentiation to other cellular populations within the blood of the Bacillus? So for that, we took a control population in our candidate hematopoietic stem cells. We labeled them non-specifically with lipophilic dye, DIO in this case, injected into young colonies and analyzed 3 weeks later this population. And what we found that while the control population, 75% of those cells stayed in the same original gate where they were taken from, our candidate hematopoietic stem cells, only 25% of the cells stayed in the same original gate, and the rest could be found on the different clusters of population on the forward scatter and side scatter, suggesting that they differentiated into other cellular population within the blood of the Bacillus. But this is a general analysis. We wanted to see how specific it is. For that, we use the pigmentation cells that we have in the blood of the Bacillus. So in our mariculture facility, we have either blue pigmentation or pure orange pigmentation or a mixture of the two pigmentation. What we took is a large donor orange colony, isolated the hematopoietic stem cells and the control populations, and we injected into young colonies. And what we have discovered is while the control population had the same ratios as uninjected cells with mixture of pigmentation, pure blue or pure orange colonies, the colonies that were injected with the hematopoietic stem cells, we had a significant reduction of the pure blue colonies. And on top of that, we had a mixture of orange and blue pigmentation cells, suggesting that the unpigmented hematopoietic stem cells differentiated into the orange pigment cells from the donor colony. So we have discovered the Bacillus Schlesser hematopoietic stem cells on the molecular level, localization, cell differentiation on a general and a specific level. But I will be straight with you. We haven't done a single analysis to prove that it's a true hematopoietic stem cells and not a population of different multipotent cells that are coming together and creating this population. Now I want to take you to something very interesting that we discovered. We wanted to analyze this hematopoietic stem cell niche in the Bacillus, the endostyle. We isolated the endostyle in different tissues in the Bacillus. We have done RNA sequencing, and we compared the genes that were expressed. And what was very interesting is that the genes that were significantly upregulated and annotated to human genes are genes that analyzed by gene analytics tool, getting the highest score is the hematopoietic bone marrow of human. And I would like to emphasize that the Bacillus I would like to emphasize that the Bacillus and humans are separated for more than 500 million years of evolution. And what we find here that there's a very high homology between our own hematopoietic bone marrow and the Bacillus stem cell niche of the endostyle. And we're not talking here about few genes, we're talking about almost 400 genes that are shared between those tissues and significantly upregulated. When we analyzed it on the mouse hematopoietic model, and this time together with the bone marrow stromal cells, we also see a significant activity of the bone marrow stromal cells compared to the endostyle. And this is something very interesting because there's nothing to do between the endostyle and a bone marrow. But when we look on the chordates and the main organs of hematopoiesis, we see that the organ itself can be very different. We talk about bone marrow in mammals and reptiles. It can be also the bursa in birds. We have head kidney in fish. In amphibians, it's changing throughout life. Epigonal organ, provertebral arc, and now we're showing the endostyle. So the organ itself can be very different. But what I'm claiming is that what is really conserved is the niche itself that is enabling the proliferation, the differentiation, and the life of those hematopoietic stem cells. And I think that studying what is conserved, that will be the key elements that we need to actually proliferate those cells and to be able to have them. And I want to say that throughout this research, we succeeded in developing the Bacillus lessori as a full model for hematopoietic stem cell transplantation, from the ability of isolating them, transplanting, interaction with the immune system, localization to the stem cell niches, and showing that they're homolog to our own hematopoietic bone marrow.
Benjamin, 1.5 minutes.
Oh, I need 2. So before I'll show you the last part and what we learned from it, I will just say that throughout this work, we have developed a lot of tools that we learn how to work with non-classical model organisms. And now in our lab, we're applying it to different models. I will just give you some examples. We succeeded in characterizing a new lymphatic network in tuna fish, characterization of, for the first time, of a receptor recombination of immune receptor in urchins. And I want to give you an example here of our work on corals and anemones. So we succeed in applying the same tools to be able to isolate different cellular populations of corals. And here you can see only with 3 different reagents, we can isolate 12 different populations, including symbionts, nematocytes, and other cellular types that we actually still don't know. But what is very interesting, that one of these cellular types is actually marked with a marker that we use for isolating stem cells, also in Bacillus, but also in other invertebrates. So from that, we have a very unique project, and the idea is stem cell therapy for corals. And bear with me. The idea is to be able to isolate stem cells from an adult coral and transplant to another adult coral. So eventually, we will be able to create this chimeric coral that will have also the phenotype of the original donor. And the idea is so we can take from a heat-resilient genotype transplant to a heat-sensitive genotype, so it will be resilient when the next heat wave will come. So I just want to give you an example of what we already started working with Nematostella. This is together with Professor Yehu Moran, who has created this Nematostella Tata box binding protein labeled with M. cherry. We used it as a donor. We transplanted it into wild type. And this is 3 weeks after transplantation. You can see that cells settled within the tissues. They proliferate within those animals. And we succeed actually in transplanting the phenotype through the stem cells. There's a lot of people that I need to thank for all this work. Of course, my advisor from the postdoc, Irving Weissman, Ayelet Voskoboinix, who's running the Bacillus project in the lab there, my collaborators, my laboratory here, my funding sources, and I would like to thank you for listening to me. And if you have any questions, now will be a good time.
Benjamin, thank you so much. It was a wonderful talk. I will ask only later and give the opportunity for others to ask. Do we have any questions? A couple of questions, of course. So I'll take the opportunity before others. I assume that you've heard of the long-term thought of Eric Davidson's about the transcriptional circuits that govern the early development of developmental programs of many metazoans. So I wonder whether not only the early metazoans are development is governed by this, but by such a program, but also differentiation to the hematopoietic system. Is it also governed by a very old and very conserved system?
This is a great question. I'll be honest with you, I do not know. We'll start with a short answer. But the longer, my assumption is that the basic mechanisms of development are probably conserved, but they change all the time. And from what we see, it seems that even the lineages within the hematopoietic tissues are different and do change through the evolution. The question is if the differentiation into the lineages, and this is something that was done also on the Botrytis genome project, showing that there is a conserve of the early stages, but as more differentiated the cells, then much less conserved mechanisms you see there. But it was not done on a specific populations yet, but more on a general lineage, but this is a suggestion at this point.
Thank you so much. Any more questions?
Thank you very much.
You're welcome. You're welcome, Benjamin. Thank you so much. It was a great talk. So for those of you who are waiting still for Jonathan Weissman to join us, he promised to join us in 50 when he starts to talk. I hope that he will not do a last minute, changes that will surprise us. But he promised that he will join. Meanwhile, we'll go to the business meeting. So some of you can take 5 and then go and do other stuff and join us later. But I need as many members as we can to stay with me. So this is the part of the business meeting. It's the most exciting part, as you might imagine. The GSI board is now comprised by myself as the president. What can I say about president? I'm the chair. I will feel better with this title. I replaced Martin Kupiec, who was the president for the past almost 10 years. Am I correct, Martin? I'm probably correct.
About 8 years. I don't remember.
8 years, okay. So I'm replacing Martin, who did a great job. Anat Ben-Zvi will continue to be on board. Judith Berman as well. Tzipi from the Galilee Medical Center. Liran Carmel has joined us as a board member. Shagi Schwartz will stay from the Weissman. And Eran Meshorer will be a controller, and along with Karen Avram. We need that, and I need you to try to please assist us and approve that. Let's do that by, yeah, let's do it by the poll that you just see. Please vote for or against the list that you just saw. Let's stay for a few more minutes. Feel free to participate. Oh, come on, just press. Let's give it one more minute and then let's go to the next thing that I would like to present you, to share with you. Okay, that's enough. I think that all of those who chose to participate in this poll chose to vote for the list of the board members. Shagi, I'm really sorry for misspelling your name. Share results. That's the results. Now, another thing that I'm obligated to share with you is how rich we are. We're extremely rich. We are extremely rich. We have 6,800 shekels in our bank account. I'm sorry, 6,600 approximately shekels in our bank account. We're gonna use them only for the next more physical meetings that we should have. But in addition to these, to the current activities, we had the idea of having additional activities. And this would be the activities. The activities are, and these are the activities, let me show you. There are, of course, the annual symposium, the annual meeting, and we thought to add some more content to our activities. And this would be, those of you who know, the annual graduate student conference in genetics, genomics, and evolution who took place for the past 10 years. It stopped for the last year due to the Corona thing. Hi, Jonathan. We're in the business.
I made it.
So don't worry.
Okay.
This annual graduate student conference, I hope that it will continue taking place with us, and I need some people to contact me later on after the meeting so we could assist you in assembling this meeting. And another two types of activities that we thought to embark on would be webinars. That this was a suggestion by Tzipi, and we need to still think what kind of webinars would be of interest for you guys as researchers, for you, your students, but also maybe for the general public. As being the Genetic Society of Israel, maybe we could share things with our thoughts and our, should I say, wisdom with the general public. So this would be not only webinars, but we thought also to launch podcasts. Podcasts cost money. We thought to add these podcasts, to fund this podcast by several biotech companies who already agreed to fund us. But what we cannot fund by biotech companies is the very existence of our society. There are several administrative things that are needed to be paid annually. And for that, we only ask you to be kind enough and maybe share with us some fees. Not a big fee, don't worry. But let's wait a moment for other people to participate in this poll. Not many people participated in the poll, come on. In a moment, I'll let you, Jonathan, to share your talk. Don't worry. Thank you, thank you, thank you. See more people participating and voting. Excellent. Yeah. So this is what people voted for. As you see, most people liked to continue the annual symposium, the annual graduate student conference, and less than half, but almost half, let me be positive about it, almost half thought that podcasts and webinars are okay. So that's what we got here. And the last thing is about the fees. So this would be the fee that we thought that if you'd be generous enough to share with us, it's not obligated this year, but it would be nice of you to share. If more than half of the participants agree, then it would be very nice of you to share with us such a fee. I'll be waiting for the vote to complete. Let's give another half a moment for people to vote. Oh, come on, be generous. Okay, so in any case, almost half of the people voted. Let me end this poll right now, share with you the results. Wow, so this means that 77% of these who voted agreed to pay the fee, and only 23% voted against. It's not an obligation, but it will be very nice of you to send us a fee, and I will share with you the method of payment for such a fee. I think that all of us will not be richer from that or less than that. So thank you so much for participating in the business meeting.
Dan, could I just say something for?
Of course you can.
Okay, I just first of all really want to thank you because and certainly Martin, who I see here, who's kind of sitting right under you, because I think that many of you know that for years, and now we're going back about 23 or so years, there was a bit of the lull in activity, and then Yossi Hirschberg came to me. I actually remember where I was at Hebrew University. Batsheva was there, you know, almost like it was yesterday, but it wasn't. And he said, come on, we've got to do something. We've got to resurrect this society. We are such a strong scientific force in this country, and many of us, I think, in the world as well. And so it was really Yossi Hirschberg who kind of pushed us to get it moving again. And I think we've had many productive years when I was there, and then Martin took over, and now you've taken over. And it's so much teamwork because of all the people who are in the committee and those that come to the conferences every year. So really, I want to thank you for, and then with the students starting their conference, it seems to be continuing except for the 1 year of COVID. So really just thank you. That's all I wanted to say.
This is thank you so much, Karen. In fact, because of you, because of your efforts, I think that this was simply resurrected. And this is the time not to only to thank Karen and former presidents. This is the time to welcome Jonathan Weissman to our conference here, the symposium. So it is a pleasure to have you here, all the way from MIT. Jonathan, can you try and see whether you are able to share with us your show, your slide?
I will.
Excellent.
Let me, sorry, shift screen.
Yes, we can all see that.
You see it?
Yes, we can. So,
all right, I just wanted to check, does it advance?
A short introduction. So, for me, Jonathan Weissman was known because of his postdocs, former postdocs. In fact, we have here one here, a very good friend of mine, Maya Schuldiner, is already here, and she's a member of the society. And we have to thank Jonathan for her work in your lab, that she joined us here. And she's not the only one who got a PI position once working under your guidance. Jonathan is well known for many genomics, introducing genomics to a wide variety of biological and genetic questions. I'm not gonna go deep into the various prizes, the amount of publications, how many citations. I'm not gonna go into that. I'm just pleased that you are here. So without further ado, please, Jonathan, the stage is yours.
Thank you so much for the kind introduction, and I'm glad to be here, at least virtually. And I'm looking forward to the time when we can all be here together. I'd like to tell you today about some recent line of research in my lab, which I think is particularly appropriate for a genetic association, genetic society, because it's work where we're able to follow the evolution of a tumor in vivo setting with, I think, unprecedented precision, and reconstruct this history. And through the combination of two technologies, that CRISPR, which acts as essentially as a writer that allows us to write information to DNA, and then single cell genomic approaches, which act as a reader that allow us to read out this information in much the same way one uses a molecular flight recorder, rather, in an airplane to be able to reconstruct the history of flight in case of an accident. So as a backdrop, and first I want to just make sure, the slide advance. Dan, did the slide advance?
Yes.
Yes.
Okay, I've had problems in the past with that, and I went through this talk, got about 10 minutes, and someone said, "You have only one slide." Anyway, so as a backdrop, one of the perhaps greatest conceptual advances, at least of the 19th century in biology, was the cell theory. And this was accompanied with a few tenets, but perhaps the most profound of those was the third one, omnis cellulae cellula, cells beget cells. And the idea that every cell comes from the division of a mother cell. And the implication is that any sets of cells have a family history, a phylogenetic tree that can connect them. And reconstructing such trees, both for cells and, as you'll see in other contexts, has been an enormously powerful tool in biology, because it lets you infer rare and distant events in the past, and then to understand their consequences. So these are familiar concepts, as I said, in biology, and applied to a wide variety of problems. So perhaps most immediate and relevant is this has been applied broadly to understand the transmission of SARS-CoV-2. So for example, one could see by taking samples of the early virus infections on the West Coast or the East Coast of the United States, you can infer that the West Coast was directly inoculated from transmission from China, whereas in the East Coast, it went through Europe, the virus mutated and then was transmitted to the US. And similarly, we can see with new variants like the B117, so-called British variant, that the rapid expansion of the tree indicates that variant is more infectious than the previous variants. So before you even do any molecular biology on the virus, study its infectivity or the mechanism, you know in a real world setting that those variants are directly linked to change in infectivity. Similarly, there's been beautiful work from David Reich and others in reconstructing the phylogenetic history of mankind as it emerged from sub-Saharan Africa and spread through the rest of Eurasia. And then on an even longer time scale, the vertebrate, the introduction of genetic sequencing, which allowed reconstruction of phylogenetic trees, has had a major impact on our understanding of vertebrate evolution, going from these sort of observational studies where you would look at which, the shape of the bones, the number of bones, et cetera, and try to infer relationships, to really having a direct inference, a direct understanding of the evolutionary relationship and the time course of this. So, for example, one of the things that's emerged from this is understanding that birds are more closely related to dinosaurs than they are to us. And then, even at a more longer time scale, one can infer from phylogenetic reconstruction that there was a single endosymbiotic event of a cyanobacterium and an aerobic bacterium separately that led to the chloroplast and mitochondria. So in the same vein, cancer is fundamentally an evolutionary process. We think that there is typically an initiating oncogenic mutation or sets of mutations, but these are not sufficient to form a true aggressive tumor, let alone to metastasize. And that in order for this to happen, you have to have a progression of genetic and epigenetic changes. And so these types of phylogenetic analysis have been used to understand where do cancers start, how do tumors spread and metastasize, and how do tumors evade therapies? How do they evolve resistance mechanisms in response to therapeutic challenge? And there have been a number of elegant studies trying to use this type of phylogenetic analyses. But broadly speaking, they fall into two classes. Prospective lineage tracing, typically done in model organisms like a mouse, where you would each clone would be clonally labeled, and then you'd be able to follow that clone as it grows and spreads. Alternatively, in work led by people like Charlie Swanton or Christina Curtis, there's been taken advantage of the natural mutations that occur during a tumor and been able to use these to start to trace the phylogenetic relationships and origins of metastases in actual human tumors. The challenge with this latter approach, though, is that the mutations tend to be rare and sparse and don't necessarily occur at the key events in the evolution of the tumor. And that, plus the challenges in recovering the tumor, limits the sort of resolution that one can get from these types of tumors. And so my lab and others, and I should say, I think a lot of this really is the intellectual grandfather of this is in many ways pioneering work from Ida Shapiro on using natural mutations and polymorphisms to follow evolution. But the idea was that rather than using these microsatellite instability, these naturally rapidly mutating regions in the genome, we could create a genomic DNA target, a scratch pad, as it were, and then use CRISPR-Cas9 to target these sites. And this CRISPR-Cas9 would target these sites slowly. They would induce a double-stranded DNA break, which then would be repaired imperfectly to generate many different indels. And then, and just a sort of a bit of a technical detour, but it's important. It's really important for our approach and how we're able to get high resolution. We use arrays of these scratch pads and then put multiple copies into the, in this case, the mouse genome, and transcribe these as an RNA so that we get an RNA copy of the information that we've recorded into our DNA scratch pads. And with these, you can now, in a very compact scratch pads, you can record in principle a huge amount of information. So if we have 13 target sites, we in principle could uniquely mark all 2 to the 40 cells in a human body, and certainly all the cells in the mouse. So just to step back and make this a little more concrete, the idea is we would engineer tumor cells, either grown in vitro or, as I'll show you, in a mouse, to have these target sites. And then slowly, during the time course of the evolution of the tumor, the cells would pick up these indels, color-coded by the nature of these indels. So there'd be a division, and one of the daughters would pick up an indel here, a second daughter here. These would then grow and divide, and the further cells would pick up additional indels. These are then transcribed as an RNA. So you would take the cells at the end of the experiment, do an RNA-seq experiment to understand their transcriptional state, basically their current state, as well as this RNA that records information about their phylogeny. You can then get an allele table. So for each cell, you know which of these scratch pads have been cut and have indels and the nature of these indels. And from this, infer lineage. So for example, the fact that cell A and cell B share this indel and cell C and D share these 2 indels indicates that A and B are more closely related to each other than C and D. And this also illustrates a very important principle used in our reconstructions and broadly used in these types of phylogenetic reconstructions, which is the principle of maximum parsimony, which is just a fancy way of saying that when you see 2 indels shared by cells C and D, you're going to assume that that's because a cell in the past picked up these indels and they both inherited them, rather than you having 4 separate events that happen to coincidentally give cell C and cell D the exact shared set of indels. And although that's maximum parsimony isn't going to be always strictly adhered to, when you have the vast sort of oversampling that's possible with these types of engineered recorders, you can be increasingly confident of the nature of the trees. So here's an example of an actual phylogenetic tree of a cancer cell growing in a mouse. This is one of our smaller trees, but helpful for illustration purposes. And you see that there was an early indel event that we infer happened really after only a couple of cell divisions that indicate all of these cells are more closely related to each other than all of these cells. An intermediate event shared by these cells indicating that they're more closely related to each other. And then a very late event that happened, in this case, only after 2 divisions before the experiment was ended, so that these 4 cells are really very closely related to each other. And this nicely illustrates the nested nature of these phylogenetic trees, so that you're able to see sort of who's your brother, who's your cousin, who's your cousin 50 times removed. And if the trees were only limited to this type of this size and complexity, you could sort of eyeball them to put them together. But our goal, and at least in some cases, what we've achieved is vastly more complicated trees. And for this, you have to go to algorithms to reconstruct the tree that best adheres to this principle of maximum parsimony. And here we've had a wonderful collaboration with Nir Yosef, as I'm sure many of you would recognize. He's an Israeli scientist from his name. Many of you, I'm sure, know him as well. But we're borrowing him at Berkeley in the computer science department. And I have a joint graduate student with Nir who worked closely with him on both the analytical and increasingly biological interpretations. And I won't go into the details of the algorithm because they've been published, but the sort of neat thing is that Nir was able to recognize that this problem of phylogenetic reconstruction actually mathematically, at least a key component of it, mathematically was identical to a problem that had already been solved, which was how to have the minimum number of wires connecting elements in an integrated circuit. And because of this, we were able to use these so-called ILP to solve this Steiner tree potential graph problem. But then, even then, our trees were too complicated, so we had to use a hybrid approach to allow this reconstruction to occur in a reasonable amount of computational time. So from this, we're able to really construct quite complicated trees. Here's an example of one involving 5,600 cells. It was 51 different recording sites and 54 days, almost 2 months of continuous recording in a mouse. 99% of the cells are uniquely marked, and the average tree depth is about 15. So we really can make very fine and nested phylogenetic relationships between the cells. And when you blow these up, they have the same sort of fractal structure where you can see those, in this case, actually multiple different indels supporting each of the layers of this tree. So that's sort of the background and the technology. And now what I'd like to do is spend the rest of the time talking about 2 applications to cancer biology. And we've also, Michelle Chen, who is a postdoc who originally developed much of this technology, has also been applying this to mouse embryology. So how do you go from a fertilized egg to a complicated vertebrate? But here I'm going to focus on 2 aspects of cancer biology. How the process of metastasis, so the process by which you have a fully formed and aggressive tumor that how it spreads through the animal with time, and then the process of how you go from oncogenic mutations, in this case, oncogenic mutation in KRAS, as well as homozygous loss of P53, one of the key tumor suppressor genes. How you go from an oncogenic mutation to develop to an aggressive tumor and then to metastasize. So you can sort of say this is sort of the final chapter of the story of the tumor, and these will be the second part will be the beginning. But so I'll start with the end, as it were. And in this, we take A549 cells, which are a patient-derived adenolung carcinoma cell line that are KRAS driven, and we engineer them with luciferase and then our CRISPR lineage tracing technology. We then inject these as a plug into the left lung, so we know where the tumors are originating, allow this to grow and spread over the course of a couple of months. Because they have luciferase at very low resolution, we can sort of follow the spreading and know when to sacrifice the mice and where to harvest the tumors from. We then harvest the tumors from different locations in the mice, and of course mark the cells by where they were harvested, so we can understand where they ended up. In the same way that you with SARS-CoV-2, you would know where you were sequencing the virus from, what the physical location, whether it be Boston, San Francisco, or Wuhan, where you're sequencing it, and then reconstruct the phylogenetic tree as well as the transcriptomic state of the single cell transcriptomic state of these tumors. And in this way, we can relate the phylogenetic tree with the final location of the tumor. And here's an example. We were actually able to reconstruct well over 100 of such tumors. And this is a fairly typical one, actually probably a little bit smaller in size than many of the tumors. But this is a single clone where a cell started out in the left lung and then grew and divided and ultimately metastasized so that it was spread throughout the body of the mouse. And we have the phylogenetic tree color-coded by the location of where those cells were harvested from. And an example here is the subtree where we have this sublineage has cells both from the mediastinum and from the right lung. And then we can infer from the structure of this tree that there was this single metastatic event where a tumor went, in this case, from the a tumor cell went from the mediastinum and then landed in the right lung and grew out to a clone within the right lung. So in this way, we can count not only how many individual metastatic events happened, how many times the tumors were able to free a cell and seed a new tumor in a different location in the mouse, but also the directionality, so the route of that metastatic event. And what we found analyzing many tumors is even though we thought this was going to be a homogeneous cancer cell line, what we found is really dramatically different behaviors in these different clones. So some were non-metastatic, that is, they stayed in the left lung where we did the initial injection. Others were distributed through the mouse, but they were relatively weakly metastatic. So this distribution came from a small number of these underlying movements, whereas others were extremely metastatic. So we would see even cells that were very closely related to each other phylogenetically, so had a common ancestor only a few cell divisions back, would be found in completely different locations within the mouse. And when you see something like this unexpected heterogeneity, one possibility that this was just incidental. It was an accident, maybe where the original cell landed. If it landed in a place that was relatively encapsulated, it couldn't metastasize. Whereas it landed next to a blood vessel, for example, it would then go to the blood and have many opportunities to metastasize. One thing, though, that we knew for sure from this is this potential to metastasize was not related to the overall size of the tumor. So the growth, the aggressiveness of the growth of the tumor in the in vivo setting was an orthogonal parameter from this metastatic. But again, was this just a stochastic event, stochastic differences, or did these really represent differences in the underlying biology of the tumor? And if you think the sort of thought experiment would be, if it's really about the underlying biology of the tumor cell, if you let the cell divide once or twice in vitro and then took two recent daughters and transplant them into different mice, then they should have a similar metastatic potential. But if it was really just an accident of where the tumor was injected, then they should be uncorrelated. Now, as it turns out, we had done multiple mice, and I should say we, it's really Jeff Quinn, a terrific postdoctoral fellow who's recently moved on to a position at Inscripta Biosciences, that because each of the tumors that we inject are clonally marked and they grew in vitro, just as I sort of said in the thought experiment, a few divisions before they were put into the mouse, we actually got two examples where the same clone was injected in two different mice. What we found is that the metastatic tree rate, the metastatic rate measured by these phylogenetic trees was dead on. In fact, for the 400 or so possible pairs between these two mice of the different clones, these two that were clonally related to each other were the closest in the metastatic score and the third closest in the metastatic score. And this would never happen by chance. So this tells you that there's an underlying inheritable heterogeneity in the tumor before we inject them into the mouse. And this then motivated trying to understand what the nature of this heterogeneity was. And as I said, in addition to reading out our phylogeny with the molecular recorder that tells us the phylogeny, we're also reading out the transcriptional state. And because we had multiple tumors for which we were measuring this metastatic potential, we could look for correlations between genes that were either upregulated or downregulated and the metastatic potential. And we found a number of such genes that were either consistently higher or consistently less well expressed in the highly metastatic tumors. And several of these genes were known factors that modulated tumors, metastasis, and invasion, but other ones either weren't known factors, or if anything, the literature would have suggested the opposite sign that we found. So a very prominent example was keratin 17, which there wasn't a lot of literature on, but to the extent that there was, it would have suggested the opposite sign. So what we then did was to take the A549 tumor cells and artificially increase or decrease the expression of these candidate genes that emerged from this analysis, and asked, did this impact the metastatic potential or the potential invasion potential of these tumor cells in vitro? And what we found is, and so we turned down the expression of the candidates with CRISPR-I or turned them up with CRISPR activation. So this allowed us to surgically sort of dial up or dial down the expression of these individual candidate genes. What we found is for 5 of the 5 genes that we tested, including keratin 17, we got the expected effect on the tumor, the invasive properties of the tumor, both for the CRISPR-I and for the CRISPR-A, and that we were able to reproduce this in a separate human lung cancer cell line, H1299 cell. So this really demonstrates that not only was there intrinsic heterogeneity, a difference in the biology of these seemingly what we would have thought was a uniform clonal cancer cell line, but that this heterogeneity was heritable and led to a difference in the metastatic potential and allowed us to identify new candidate genes. So this is just, I guess, reiterating what I said, that but maybe I'll add one final point, which is we found these distinct metastatic states that they were in general stably inherited. So within the clone, typically all the cells were either highly metastatic or lowly metastatic. But we found a couple of examples where the tumor was actually evolving, so it was sort of caught red-handed, evolving so that one of the branches was low metastatic and the other became highly metastatic. And we could then again see through the transcriptomic analysis what were the drivers of this change. So with that, I'll finish with an even more recent story in the lab, which is work led by Dian Yang with a lot of help from Santiago Naranjo, and also in a close collaboration with Matt Jones and collaboration with Tyler Jack's lab of studying tumor initiation. And here we use an elegant model for tumor evolution developed by the Jack's lab, the so-called KP model. And this is engineered embryonic stem cells, mouse embryonic stem cells that have oncogenic KRAS mutation and potential loss of P53. But at the baseline, neither the oncogenic KRAS nor the loss of P53 is present, but they can be induced by a Cre recombinase. In addition to these, so this KP method Tyler's used extensively, we engineered in our lineage tracing system, again, with a Cas9 that has a lock-soft lock. So no tracing is occurring. The mouse is born, develops normally, and then as an adult, we deliver through inhalation lentivirus containing the Cre recombinase. And so what that does is in individual cells, which we separately barcode, it initiates both these oncogenic mutations and the lineage tracing system. We then allow these tumors to evolve and grow into aggressive tumors, into metastasize, and then at the end of the experiment, harvest the tumors, do the phylogenetic analysis, compare this to the transcriptional state, and reconstruct the evolutionary history. And this is really, I think, a very natural application of our lineage tracing, because some of the key questions that had been defined in the field were immediately addressable by this approach. So things like, how do tumors expand and what drives their expansion? What is the origin, hierarchy, and transitions of transcriptional heterogeneity during tumor evolution? And what is the origin, hierarchy, and molecular mechanism of metastasis? So on the one hand, these are really very nice lineage questions. On the other hand, they're questions about the current cell state, and this ability to relate them is really the power of the system. So before that, I'll do a population genetics 101, how to think and interpret these phylogenetic trees, even before you get into the molecular nature of differences in the cells in these trees. And broadly speaking, we think of if there's neutral evolution, some branches might outgrow others just by chance, but there's limits to how much this is going to happen. So you would a sign of neutral evolution is relatively balanced trees, whereas if you start to get a positive selection and a selective advantage for elements of these trees, that branch will expand, and you can see this either as a single event or as a branched event. And so we're gratified when we start to analyze the phylogenetic trees from tumors from this KP mouse model. We saw for some of our early tumors that they were relatively balanced, although if you do the actual analysis, you can start to see evidence for a positive selection. But in other tumors, you were starting to see really very clear evidence in positive selection. And then at later times, really seeing clonal sweeps, either in this case, a single clonal sweep, or in this case, two independent clonal sweeps. And so now we're really in a position to try to understand how these sweeps happen, what's the underlying molecular nature of them, and the routes by which they occur. And just we can go from this qualitative picture to a more quantitative picture of this by essentially for every cell analyze the degree of expansion of the branch it's in, and from this and only from the structure of the tree, not looking at all at the molecular nature of the tumor itself at this stage at least, we can assign a fitness score. And from this, we can now relate it to the underlying transcriptional state of the cell to understand what are the drivers of this change in fitness, this drive to be an expanding tumor. And we can then overlay the earlier work, single cell analysis, that had defined the different transcriptional states that these KP tumors can exist in. So this is an amalgam of single cell RNA-seq of multiple different tumors from these KP mice, where you define multiple distinct transcriptional states. But at least from this, have no idea of how you go for. We know that it starts in the AT2-like state and ends up in things like these extreme EMTs, these highly aggressive tumors, but really have very little direct evidence from this alone on how you get from A to Z. And what we're able to do now is to overlay these, because we have the phylogenetic data, which previously had not been available, we can overlay the fitness and so see how this evolution from a relatively benign, slow-growing cell to an aggressive tumor occurs, and you can define distinct modules, distinct pathways by which this conversion to an aggressive tumor can happen. And so just I'll finish here with sort of bringing these pieces together and how we can study the evolution of tumor and its ultimate metastasis, and the first inklings of how this can be integrated with spatial information, because of course the tumor doesn't grow in a flat dish; it grows in the mouse and with defined stromal influences as well. So what John did was to take these tumors and sort of do poor man's spatial genomics. He literally cut it into 15 different pieces and then did a multi-regional analysis of the phylogenetic tree. And what he saw, and then overlaid the spatial location of the tumor with the phylogenetic tree. And first of all, what he saw was that there was very nice coherence. So different regions of the tumor came from different phylogenetic branches. That's satisfying and certainly gave us extra confidence in the integrity of our phylogenetic reconstruction. So you see the 1 to 4 region came here, 5 to 10 came from these subtrees, and the 11 to 14 came from these subtrees. But you'll also notice that there's some of the branches are missing. They weren't in this tumor because they were actually harvested from different metastases. And we can now see that, first of all, that there was 2 clonal sweeps in this tumor, and that one of them gave rise to the soft tissue metastases, and the second, in 3 independent events, gave rise to these 3 different liver metastases. So the picture then emerges is that the metastases arose late from defined physical and phylogenetic locations of this tumor evolution, and with 2 distinct events, one that was prone to giving liver metastases, another to soft tissue metastases. So just to sort of summarize a little bit of what we've learned and what I talked about, I showed you that we can use these phylogenetic reconstructions to measure the rate of metastases and find expression signatures that allow us to identify drivers of metastases. And work that I didn't get a chance to talk about, we've used this also to follow the roots and directionality of the metastases. So how it goes from one location, the left lung, to the mediastinum, and then to the rest of the body, identifying the mediastinum as this sort of way station from which once the cells go, they can then have access to the rest of the mouse. And then finally, I've used this show to talk about how we can monitor progression from an oncogenic mutation to aggressive and ultimately metastatic tumor. And so what are some of the future directions? So what I have not talked about is the sort of last stage in the tumor, at least is that, of course, is the attempts to treat it, and both by targeted and our immuno-oncology approaches to treat tumors and the origin of resistance. So which populations survive, and are there parallel genetic routes for chemotherapeutic resistance? I showed you a very low resolution spatial information, but I hope enough to show you that, to convince you that there'll be much more there if we can get higher spatial resolution. Unfortunately, there have been a number of approaches that allow much higher, even single cell spatial resolution. And so we're very interested in overlaying this both to understand the spatial structure of the tumor itself, as well as its interaction with the stroma. And then finally, I've talked about a molecular recorder and the idea of recording different events in the life history of the cell. But in fact, all I really presented was this phylogenetic reconstruction. But in the same way we have these DNA scratch pads that are marking the phylogeny, we can hook up the activity of Cas9 to other molecular events so for example, hypoxia or interaction with the immune system or inflammatory signals, and have these events recorded in these DNA scratch pads and overlay then these richer history of the cell with its current state. So with that, I'll thank you all for listening, and want to acknowledge the people, the major players in this. I've talked about them some along the way, but Jeff Quinn and Matt Jones and Ross Okimoto were key players in the metastatic story. That's been a wonderful collaboration with Trevor Pavona at UCSF and Nir Yosef at Berkeley. And then Dion has really been leading this tumor evolution in the KRAS P53 model. And that's been a terrific collaboration with Tyler Jack's lab. So I'll end there and take any questions.
Thank you so much, Jonathan. This was wonderful. We are slightly over time, so I think that we have time for 3 questions in addition to mine. So I'll take the opportunity and ask the first one. You listed, you did a wonderful evolutionary genomics work with a single cell analysis here. And I wonder whether you could use this information to predict, before you do the experiments, to predict which of the clones that you managed to isolate would survive better in hypoxic conditions or in non-hypoxic conditions, in other environmental, body environmental conditions or not. I assume that you're gonna do the experiments, but can you use the transcriptomics to predict?
Yeah, no, this is a terrific point, and I think there's sort of 2 ways to do this. What's getting at is there's all sort of inferential. You see a change in transcription shared by cells, and then you see a shared change in behavior, and you therefore infer that that change in transcription or change in state of the cell was responsible, but it could be incidental. And so there's sort of a couple ways to do this. One is by a serial. So you would take out cells, analyze them, and then put them back in the next mouse. And although the analysis is destructive because you would have sister cells that could be transplanted, you could then watch how they behaved. And then the other, of course, is as we did, we turn up and down candidate genes and see their causal impact. So I do think there is, this is really very much a hypothesis generating approach, but I think it can lead to very concrete ways. Now, I'll say one other thing, just we'd love to be able to do though, is to do this in a non-destructive way. So we'd like to be able to reconstruct the file, watch the phylogenies or watch the information that we were recorded without having to, sacrifice the mouse and put the cells through a 10x genomics. And that's going to be hard, but one thought of how to do this was if you could have the tumor cells emit a viral-like particle in a defined way that gathered the informational RNAs, you could then do sort of a liquid biopsy and get the state of those recorders at that time. That's a long-winded question. I'm not sure if I answered it, but
Thank you so much. So we have a couple more questions. Maya, you'll be the first.
Hey, Jonathan, great to see you. I have a question about the technology itself. So it seems like you have tens of thousands of events of repair of double-strand breaks, and you also have the lineage tracing to see which one occurred once and which one occurred over multiple times. So can you use it to say something about actually the DNA damage repair machinery and its tendency to fix double-strand breaks in specific ways? And if it's random, that's fine. But if it's not random, do you think that would have create biases in your data? And I guess a sub kind of then question of that is, as tumors have mutations in repair machinery, do you see that I mean, you could correlate it because you know the exact moment potentially, yeah, they had mutations in their DNA repair machinery, and you could correlate that to changes in how.
Yeah, so these are broadly, if I understand sort of the question, are one, maybe you didn't say it this way because you're being polite, but one is our recorder, because it's causing double-strand breaks, changing the biology of the tumor itself? And then the second is, could we actually use this to try to understand how the tumor is responding to DNA break? What I would say is the double-strand breaks absolutely can cause challenges. Although each individual cell has a limited number you can get a huge amount of information out because of combinatorial and because we chose, this is a little in the weeds, but we chose sites that had a broad indel distribution. So if they all went to single nucleotide insertion, then 2 cells that had that single nucleotide insertion could have happened by chance. But if there's 50 different outcomes, which we have in this case, then it's much less likely that they had by chance. And that lets you get away with fewer breaks, essentially. The short answer is the breaks definitely can be a problem, especially in the embryogenesis, where when we had too many breaks and we're trying to record things quickly, we had problems with viability, although we were able to get live and seemingly normal mice out of it. The p53 null nature of the tumor, I think, helps a lot. They sort of just burn through these breaks. And also, we're recording, although we might have dozens of breaks at the end, we actually cripple the guides so that they occur slowly, so that they occur over the course of a couple of months. I would say the fact that you can take sort of the 2 cells and go into different animals is another argument that it's not having a big impact. But mostly we've worried a lot about this, and we're thinking about things. Can we use things like base editors or things that would not cause double-stranded breaks as a way of avoiding this? But I think at least for the tumor, it's probably, compared to everything else that's happening genomically, in a bit of a wash.
Thank you so much, Jonathan. We have a couple of more questions, and please, quick question, quick answer. Judith.
Okay, I would like to know whether you have any idea what underlies the differences in the transcriptional regulation of the new metastatic, the different metastatic programs.
The short answer is no. And the longer answer would be, we know it's a pre-existing diversity. So what we should do is try to fish out the cells that when clones of the cells that when put in have different metastatic properties, and do more deep genomics on this. So do things like single cell DNA sequencing. This is possible to do, for example, from the KP mouse. You can take a subset of your tumor cells and do deep sequencing of their DNA or of their chromatin state, and then put them on the phylogenetic tree to understand what their behavior is. But we just really, what we've done is some copy number variance analysis and transcription analysis, but we don't know to the extent it's epigenetic versus genetic differences. All right. Well, if there aren't any other questions.
I have a question. I think, Dan, you were on mute. Yeah. Short question. Great talk. Thank you. To what extent can this deal with copy number changes and rearrangement in the genome, which we know are all essential part of cancer?
I'll start with the first, which was a technical, which is if you get a copy number change in your lineage recorder itself, it's a bit of a pain because you now get it incoherent. You get some incoherence, and we see this, and we basically, the recorders are distributed. So we have 13 different sites. So if we get a copy number variant change, and so 2 marks at the same site at what we think is the same copy, we tend to just throw it out, but I think we could use it better. The other is, so by looking at the transcriptomics, using a very nice algorithm developed by Aviv Regev, that we can, you can infer a copy number variation, and we can actually see as the tumor becomes more aggressive, it becomes more prone to generating CNVs. So you basically look for contiguous regions that have higher or lower expression and infer that there was an underlying CNV.
Thank you so much. Thank you so much, Jonathan. Thank you, Ron, for asking the last question. And I want to thank all the speakers that were brave enough to give their talks in this terrible medium. I'm sorry. So we'll hope to meet all together physically next year. And I really thank you all for attending. Thank you, Jonathan, for keeping for being with us despite the time differences. And thank you all
Yeah.
for attending.
Thank you,
Oh, it's
Jonathan.
my pleasure.
Good to hear you.
Don, can I give a shout out for a meeting that we have coming out?
Go ahead.
Okay, because there's several people in the audience here who are involved. So the Human Hugo, we're having a virtual meeting on March 22nd. We'll be sending out an announcement. Hopefully Don will be kind enough to send it out to all of you. And this is really the virtual predecessor to the in-person meeting, October 4th through 6th, hopefully in Tel Aviv live. And we have a really fantastic crew that's going to be speaking on genomics. Several of you in the audience are already invited and have confirmed your participation. So I would really appreciate it if you could join us. Thank you.
I second to that.
Do you repeat the date?
March 22nd in the afternoon at 4 p.m. is the virtual meeting for about an hour and a half. And then October 4th through 6th in October. And I'll ask Dan to send out a notice and you can get all that information.
Thank you.
Thank you.
Okay, guys.
You have to have the last
Thank
word.
you very much.
Bye, everybody.
Thank you.
Bye-bye. Thank you for joining.